We are at the dawn of a new era in genetic technologies. Technologies that have really been developed over the last 4 or 5 years that are going to affect us all. That's us, our children, and our environment. Everything living has the potential to be affected by this technology. So this is a 5-minute video based on non-scientists or aimed at non-scientists for CRISPR Cas9, the new genetic technique. So what is CRISPR Cas9? CRISPR Cas9 is a system that was first observed in bacteria, and it was part of their immune system. Who would have thought bacteria also have immune systems? And they use it to identify viruses that infect them. So if viruses infect a bacteria, they would use this system to cut them up using an enzyme, a Cas9 enzyme, to make them inactive. And that basic technology has been switched and used and can be utilized and applied to higher organisms, higher animal cells, and humans. So what's new about this technique? Well, up to very recently, maybe even 10 years ago, we had a problem for genetic manipulation. You are no doubtedly aware our genome is huge, hundreds of thousands of genes. And the problem is, if we wanted to isolate one of those genes or look at one of those genes or introduce something into our genome, we had no real control of where things went. So let's give you an example. Say there was a disease and we knew one gene causes a disease. Let's say sickle cell anemia, something like that, or Huntington's disease. In principle, we may have been able to repair that gene, but we never had a system that was targeted enough to get to where we needed it to be. Anything that we would introduce or cut would happen at random. It was very difficult to be precise, and that is exactly what CRISPR Cas9 allows us to do. It allows us to choose exactly where we want to go and cut or manipulate in the genome. And that applies to anything from, as I said, from amoeba to humans, chimpanzees, mice, any higher cell. And that's what makes it so powerful. So let's go into just a little bit more detail of how CRISPR Cas9 works. So what does it stand for? CRISPR Cas9 is clustered, repetitive, interdispersed, palindromic repeats. I'm not going to go there. But what it does is, in a nutshell, is that you can design small bits of DNA, which is incredibly easy to do, to targets that are complementary to the place we want to go into in our genes. So it can go anywhere in theory. And then the Cas9 is actually a cutting tool. So you can get wherever you want to, and then you can cut it. So if you know where the gene is that you want, or you want to find and you want to replace it, target it, you would just design a little complementary piece of DNA. It would go to where you want in the genome, cut. So there you go. You can manipulate genes specifically. But what you can also do is, when you cut this, it gives us the ability also to replace genes. You can either cut them to deactivate genes, stop them working. If you want to know, for example, what a gene does, you might want to cut it and stop it working, and then you will have a look at, see what the effects are. But what you can also do is, if you know that there's a faulty gene, the theory is that you can actually cut out the faulty gene and then replace it with a new one. It is that precise, and that is why people are so excited about this technology. It gives us unrivaled capacity to change our genes.